Hello students, welcome to EPG Pathshala. I am Dr. Rabia Parveen from Jamia Hamdard. Today we will discuss in vitro testing of cosmetics for safety. In this, we will discuss about the safety concerns of cosmetics, then safety evaluation of a cosmetic product, their in vitro testing, chemical test for cosmetics, microbiological test for cosmetics, and then conclusion. First, we will start with the introduction. You should know what is cosmetic. The word cosmetic has been derived from cosmeticos, that means skill to decorate. Therefore, cosmetics is the branch of pharmaceutical science that deals with the art of decorating yourself. According to Drug and Cosmetics Act, cosmetics are defined as particles intended to be rubbed, sprinkled, poured or sprayed on, applied to or introduced into the human body or any part or organ of the body for the purpose of beautifying, cleansing, to promote attractiveness or to alter the appearance. Cosmetics are applied on skin, nails, eyes and other parts of the body, and on this basis cosmetics are divided into different categories that you can see on the screen. Cosmetics like toiletries, hair care products and skin care products frequently cause skin allergies or other adverse reactions like allergic contact dermatitis, and it is the commonest single reason for hospital referrals. It is reported that 13% of the total Indian population are allergic to those cosmetics. From the dermatological point of view, the cosmetics can be categorized as skin care products that include moisturizing agents, whitening agents or cleansing agents. Then hair care products that include all type of shampoos and conditioners, hair styling agents and hair dyes. Then face makeup products that include face powders, foundations, rouge, concealer, blushes, lipsticks, mascara, eyeliners and eyeshadows. Then nail products that include cuticle or nail cream, nail colors, nail color removers, etc. Then perfumed products includes antiperspirants, deodorants, perfumes, food spray and aftershaves. Then dental products that include dentifrices, toothpaste, gargles, mouth paints and mouthwashes. And the last category is the miscellaneous cosmetics that include sunscreen preparations and anti-aging preparations. Now come to the safety concerns with cosmetics. Adverse reaction with cleansing cosmetics are very rare because they are applied on the body and remain in touch with skin for a very short duration of time, but perfume or deodorants like products may cause allergic reactions and skin irritation. Moisturizing agent increase the hygroscopic properties of skin, but higher concentration of such substances may cause exfoliation and irritation of the skin. Depigmenting agents or skin lightening cosmetics containing hydroquinone, most commonly prescribed agent that cause ochronosis and mutagenicity. Ochronosis is characterized by darkening of the skin area to which formulation containing high amount of hydroquinone is applied for long term. Several reports are available in the literature of immediate allergic reactions and also known as anaphylactic reactions after application of henna dyes. Other than skin reactions, sneezing, running nose, dry cough or shortness of breath can be an adverse reaction of cosmetics. The sunscreen preparations may cause skin irritation, photoallergies or phototoxic reactions due to the excipients like stabilizers or fragrances added to the cosmetic and not due to the presence of active ingredient. Benzophenones are such an examples that cause irritation, and dibenzoyl methanes, cinnamates and para-aminobenzoic acid may cause photoallergic dermatitis. The adverse reactions associated with antiperspirants, deodorants, perfumes or fragrances are due to the fragrant material or some other ingredients present in it. Fragrance may enter the body either by skin ingestion or via the lungs or through nose. It reaches directly to the brain. This can result in headaches, dizziness, irritation to nose, eyes and throat, fatigue and other symptoms. The ingredient that is responsible for aroma is the most common reason of skin adverse reactions to cosmetics. airborne contact dermatitis is the skin reaction that is produced due to the presence of perfumes. Other additives like methyl eugenol are suspected as carcinogens. Shampoos or conditioners rarely cause cutaneous irritation or allergic dermatitis, but eye irritation may be a problem. Common sensitizers added in shampoos include formalin, hexachlorophene, parabens, triclosan or fragrances. Scarring alopecia and hair shaft breakage are the adverse reactions associated with straightening agents with pressing oils and round tongs or heated metal combs. Skin irritation, ingrown hair, and cuts in the skin are the adverse effects associated with shaving preparations. Hair bleaching products contain hydrogen peroxide as the main ingredient that oxidizes melanin present in the skin to a lighter color. Temporary skin discoloration, skin irritation, pruritus and the prominent bleached hair against naturally dark skin are the disadvantages with bleaching agents. Ammonium persulfate are also added in hair bleaching products that can cause type 1 and 4 allergic contact dermatitis. The other adverse effects associated with ammonium persulfate are asthma, generalized urticaria, syncope and shock. With the eye care products, around 12% of adverse reactions occur on eyelid, that is due to application of eyeshadows. Eye care product cause irritant contact dermatitis instead of allergic contact dermatitis. The most commonly used eye makeup product is mascara, and the most feared adverse reaction of it is bacterial infection by Pseudomonas aeruginosa in corneas that can permanently destroy the visual acuity. Surma or kajal contains carbon compounds and also heavy metals like lead or mercury that might cause serious adverse reactions. The common dermatological reactions with the use of nail varnishes include allergic contact dermatitis and nail plate discoloration. Dissolved pigments can cause nail staining problem also. The adverse cosmetic reactions may occur either due to the presence of active ingredient or due to the other additives present in the cosmetic product. The skin reaction might be occur due to any procedural error or contamination of a final product. As we discussed above that the fragrances are the most common cause of adverse reactions, the second most common cause of such reactions is the addition of preservatives. Preservatives generally cause irritation instead of allergic reactions. Only mild effects occur like smarting, stinging and contact urticaria. In some cases, serious reactions may occur with edema, redness, dryness or scaling. Therefore, it is necessary to conduct safety tests before human use. The adverse reactions due to application of cosmetic products may be in the form of acute toxicity, skin irritation, percutaneous absorption, eye irritation, photosensitization and skin sensitization, mutagenicity or genotoxicity, subchronic toxicity and phototoxicity or photoirritation. The safety test should be carried out for test for hypersensitivity reactions, test for irritancy or sensitivity, test for occlusivity, test for phototoxicity and test for genotoxicity or mutagenicity. Now come to the next heading, that is safety evaluation of a cosmetic product. The safety assessment of cosmetics depends on how and on which area it is applied, because amount of cosmetic ingredients that may be inhaled, ingested or absorbed from the mucous membranes or the skin depends on type and route of administration. The optimum concentration of active ingredients and other additives is also important. Too high concentrations and a long-term use produce false positive effects because of their sensitivity and irritant effect, whereas too low concentrations result in false negative reactions. The factors on which the safety assessment of cosmetic depends are site of application, how it is applied, quantity of active ingredients and other excipients, and the duration of time of use. Now come to the in vitro testing of cosmetics. Several toxicological studies or other in vivo studies are available for evaluation of safety assessment of cosmetics. But since March 11, 2013, the 7th Amendment to the European Union Cosmetics Directive banned the sale in the European countries of any cosmetic preparation that itself tested or contains additives that were tested using laboratory animals, thereby effectively banned the use of laboratory animals for testing of cosmetics and their excipients. Other countries also banned the animal testing models for topical products. For local toxicity and genotoxicity, only few in vitro methods were developed as an alternatives to animal testing methods. The safety assessment of personal care products has been reviewed. They are applied topically to human skin and produce local exposure, but after skin penetration they produce systemic exposure also, and if used in oral cavity, on face, eyes, lips or on mucous membrane may also produce systemic exposure. In some countries like Europe, America and Japan, the safety assessment of personal care products is regulated under Regulation for Drug and Cosmetics. For in vitro techniques, attempts have been made for developing computer-based modeling techniques to provide predictions about chronic toxicity or repeated dose toxicity. But at present, only few such models are available. The omics technology has been applied to in vitro models for understanding and predicting the toxicity produced by cosmetics. The methods are available for identifying the adverse reactions of substances but not appropriate for quantitative safety assessment of cosmetics. The in vitro tests are divided into three types that you can see on the screen. First is physical test for cosmetics, then chemical test of cosmetics, and then microbiological testing of cosmetics. Now come to the common in vitro tests that are used for in vitro testing of cosmetics for safety. First is percutaneous absorption test. You should know what is percutaneous absorption. It is defined as the movement of a cosmetic ingredient applied onto the surface of skin to the circulatory system. The absorbed dose after percutaneous absorption is the amount of an ingredient or a drug that is systemically distributed. If an ingredient of a cosmetic is found to be penetrated via the stratum corneum into the deeper layers of skin, then it should be considered as absorbed. The percutaneous test is carried out to estimate the absorption rate of a cosmetic ingredients across the different layers of skin. The absorption takes place in three steps. First is penetration of ingredient into the layers of skin layer, then penetration from one layer to skin to another, then resorption into the vascular system. In vitro and in vivo models are available for safety assessment of cosmetics, but due to the ethical and feasibility reasons, in vitro models are more preferred over in vivo models. For percutaneous absorption, transdiffusion cells are used for study the drug release pattern. Some specific solvent is used at the receptor compartment. The formulation is applied on the surface of cellulose acetate membrane. The membrane will then clamped between the donor and the receptor compartment. The donor compartment is kept in contact with the receptor compartment and the temperature should be maintained at 37 degrees Celsius. The diagram of transdiffusion cell you can see on the screen. In this, the solution in the receptor compartment should be continuously stirred by using magnetic stirrer, and at predetermined time intervals a small volume of solution from receptor compartment is taken out and immediately replaced with same volume of the solvent or solution to maintain the sink conditions. After suitable dilution, the drug concentration in the receptor fluid will be determined by UV spectrophotometer or other analytical techniques like chromatographic methods against appropriate blank solution. Now come to the next test, that is chicken eye test for ocular occlusivity. You can see on the screen schematic representation of chicken eye test method. In this, the isolated chicken eye test is an in vitro method that is used to classify the cosmetic ingredients as severe irritants or ocular corrosives. This method uses chicken eyes obtained from slaughterhouse where the chickens are killed and used for human consumption, thus eliminates the need of laboratory animals. The eye should be inoculated and is mounted in an eye holder with cornea positioned horizontally. The test ingredient and the positive or negative controls are applied to cornea. The toxic effects in cornea are observed and measured by a qualitative assessment, qualitatively in terms of opacity, damage to epithelium that is based on fluorescein retention, and quantitatively by measuring the increased thickness, that is swelling. Next test is Headcam for irritancy potential. The most important biological property of a consumer products is the local compatibility to skin or mucous membranes. In vivo tests are available to estimate the skin irritation potential of cosmetics and additives present in the cosmetic formulation and suitable for risk assessment. The Headcam, that is hen's egg test on chorioallantoic membrane of fertilized chicken eggs, is used to determine irritancy potential of the skin that you can see on the screen. This method was validated by the COLIPA, that is the European Cosmetic, Toiletry and Perfumery Association, as an alternative to Drey's rabbit's eye test, that is in vivo test. Before COLIPA validation study, Headcam was pre-validated with 100 test ingredients with different physical appearance and chemical structures that are used in cosmetic industry. There is a high concordance of Headcam results with the data of in vivo Drey's rabbit's eye test, especially for the slightly irritating test ingredients. Based on this, Headcam test is now taken as a standard operating procedure in COLIPA validation study. The Headcam test has advantages like robustness of the method with good prediction of skin irritation potential. The test include 10-day-old fertile eggs, which are divided into 6 test groups, 3 test ingredients concentrations, 2 positive controls and 1 vehicle control group. The eggs are placed in incubators. On day 10th, the eggs are removed from incubator and candled to determine the viability of embryo. A rectangular window is removed from shell directly and the egg membrane is carefully moistened with 0.9% saline and then placed back to the incubator. The eggs are dosed and observed for 5 minutes for appearance of any change like lysis, hemorrhaging or coagulation. Now come to the next test, that is skin sensitization studies. Skin sensitization studies are used for the safety analysis of cosmetic ingredients. The method involves model peptides standardized for groups of excipients with wide-ranging reactivity reaction mechanisms. Results in adduct formation. In vitro integrated testing strategy is a series of tests used for skin sensitization that you can see on the screen. In this, first is the in vitro sensitization assay. It is the test of keratinocyte activation. It uses an ELISA method for measuring the interleukin, that is IL-18, released from topically applied reconstructed 3D human keratinocyte tissue model. And next test is human cell line activation test. It is a test to determine dendritic cell activation. It involves flow cytometric analysis for measuring the expression of CD86 and CD54 on treated THP1 cell culture. Now, third is direct peptide reactivity assay. It is an in chemico method for prediction of epidermal protein binding. Binding of epidermal proteins is a measure of skin sensitization. This method uses HPLC analysis for measuring the depletion of synthetic peptides in solution form after exposure to test ingredients. Now come to the next in vitro test for cosmetics, that is direct peptide depletion studies. In this, set of ingredients are brought in contact with specific nucleophiles containing peptides like cysteine, lysine or glutathione. Quantitative analysis is based on disappearance of target peptide or the percent depletion of the peptide, whereas qualitative determination is based on formation of covalent adducts with peptide. An aspect of chemical reactivity provide a basis for measuring the skin sensitization potential. The formation of covalent adduct is considered as an indication of potential allergenistic reaction of the test ingredient. The test of ingredients are rapidly screened by LCMS method, and the stepwise procedure of the method you can see on the screen. Now come to the next test, that is test for skin moisturizing capacity. The tissue engineered human skin 3D model is used for testing of cosmetic formulations and the raw material used in skin cosmetics. The 3D model is prepared by reconstructing the skin using collagen, glycosaminoglycan chitosan bipolymer. The dermal fibroblast and human keratinocytes are co-cultured into the skin to create a tissue which closely resembles in vivo architecture of normal skin. This can be used to study the complex interactions between dermis and epidermis, and also separate responses of epidermal and dermal layers can be assessed. This 3D model have certain advantages like long-term cultivation is possible with repeated application of cosmetic formulation containing active ingredient. Similar architecture as of normal human skin to determine the interaction between epidermis and dermis. The similarities include expression of keratinocyte differentiation markers like cytokeratin 10, transglutaminase and filaggrin, and some proteins of the basal lamina like collagen type 4 and laminin, and extracellular matrix proteins like elastin. The efficacy of some specific bioactives can be determined by different endpoints like stimulation of collagen synthesis in dermal layer, stimulation of filaggrin responsible for skin moisturization. Now come to the next in vitro test, that is in vitro mammalian cell chromosome abrasion test for genotoxicity. The in vitro mammalian cell chromosome abrasion test is carried out to assess the genotoxic potential of test ingredient. Mammalian cells are cultured in vitro, then exposed to test ingredients, harvest the cells, and frequency of chromosome abrasions with asymmetrical structure is measured. Procedures should be carried out very carefully to obtain accurate results and to avoid false positive results. The purpose of this test is to identify the ingredients that can cause structural chromosome aberrations in cultured mammalian somatic cells. Aberrations can be of two types. One is structural chromosome aberrations or the structural chromatid aberrations. In vitro uses cultures of established cell lines, primary cell cultures or cell strains. Cells are exposed to the test ingredient. It can be either solid or liquid, or both with and without metabolic activation during 1.5 normal cell cycle length. At least 3 concentration of test ingredient should be taken, and each concentration duplicate culture should be used. At predetermined time intervals after exposure to cell to test ingredient, the cells from the culture are treated with metaphase arresting substance, then harvested and finally stained. The cells in metaphase are analyzed for the presence of chromosome aberrations by microscopic techniques. Now, after in vitro chemical testing, come to the chemical analysis of cosmetics. Chemical analysis of cosmetic formulation is an important to check that the cosmetic formulation contains only the permitted ingredients and the information on the label is correct or not, and also helps in forensic investigations. The concentration of the chemical compounds or the metals should be optimum for safety point of view. Different types of excipients are added to different cosmetic products, and the concentration of metal ions are measured by several analytical techniques. First, chemical testing of deodorants and antiperspirants. These contain aluminum zirconium tetrachlorohydrate glycine complex and aluminum chlorohydrate as the main ingredients. The other additives in these formulations are fragrances or perfumes and bacteriostatic or bactericidal agents like triclosan as preservatives, buffers like triethyl citrate, or the substances that can bind with offending smell are also added to the cosmetic products. Aluminium compounds are taken up via hair shaft or through pores in the skin and react with water to form a gel that blocks the perspiration. The perspiration releases toxins from human body, and if perspiration blocks, the toxins get accumulated in the lymph nodes for years and may cause breast cancer. Therefore, detection of these metals are important. The concentration of zinc and aluminum in deodorants is determined by flame atomic absorption spectroscopy or the gravimetric method. Now, the zirconium ion that is also present in deodorants or antiperspirants. The zirconium ion concentration is detected by flame atomic absorption spectrometry or colorimetric method. Boric acid present in deodorants can be detected by ion exchange method. The concentration of chlorides and sulfates in deodorants are determined by gravimetric method. The concentration of methenamine and urea added in deodorants are determined by titrimetric methods. Now come to hair care preparations. Different types of hair can be benefited from different treatment approaches to promote length and growth of hairs. External factors like exposure to UV rays of sun, dirt and smoke, dietary factors like deficiency of vitamins and essential fatty acids, and the additives in hair care products when applied to hairs and scalp and other hair treatments alone or together can cause problems to existing hair that results in hair growth impairment. Additives used in shampoos or in other hair care products include surfactants, antimicrobial agents and preservatives that can impair the different aspects of hair health. Therefore, the safety assessment of some ingredients in hair care products and helps in providing possible nourishment for improvement of hair health. The presence of quinine in shampoos or in hair lotion can be detected by HPLC. The presence of alkaline salt and oxalic acid in hair care products are detected by filtration process. Potassium hydroxide and free sodium can be determined in hair straightening products by filtration. The quantitative and qualitative determination of mercaptoacetic acid in hair waving and in hair straightening products is carried out by GC or iodometric titrations. The concentration of selenium disulfide in terms of selenium in anti-dandruff shampoos can be determined by atomic absorption spectrometric methods. Now, chemical testing of toothpaste. Fluoride containing tooth powder or paste produce acute toxicity if swallowed in high concentrations. Vomiting or nausea can be the problems related to fluoride ingestion on topical application. The sweeteners used in dental products are toxic in nature, for example, diethylene glycol. The active ingredient of many types toothpaste is triclosan, which when combines with chlorine present in tap water forms chloroform, and chloroform is reported to be a carcinogen. Sodium lauryl sulfate is used in toothpaste to produce foam on brushing can alter the taste perception. Therefore, keeping in mind the safety of dental care products, analysis of such ingredient is very important. The presence of chlorates of alkali metals and chloroform present in dental products is detected by gas chromatography. The concentration of total fluorine in dental creams or paste is determined by gas chromatography. Now come to chemical testing of creams and paste. Due to addition of preservatives in creams, they show a significant risk of immunological sensitization. Ointments are moisturizing, suitable for dry skin, and have a low risk of sensitization and irritation. Gels cause significant risk of hypersensitivity reactions due to presence of preservatives and fragrances. Therefore, the quantity of such ingredients should be known and the formulation should be analyzed before human use. The qualitative and quantitative analysis of paste and nitrite creams are done by colorimetric methods. The quantity of water in vanishing cream is determined by Karl-Fischer titrimetric method. The ash value of vanishing cream is determined to check the total organic content present in it. The chloroform soluble compounds are detected by gas chromatography. Now come to the microbiological testing of cosmetics. As cosmetics are applied on the body parts, so there should not be any microorganisms present in it. The detection and removal of microorganisms from cosmetic product is important to increase the shelf life of the product and also ensure quality of a product, consistency and overall performance to meet the federal regulations. To improve the quality and safety of cosmetic products, it is very important and necessary to carry out routine microbiological testing or control on routine basis from every batch of finished cosmetic products before market approval and human use. Generally, the microbiological testing or controls comprise the counting of bacterial cells if present in the finished cosmetic products and testing of efficacy of preservatives at different levels. Cosmetic products contains active ingredients as well as different types of additives which are recognized as the substrates for the growth and survival of huge variety of microbes. These ingredients possess types of nutrients like polysaccharides, lipids, amino acids, proteins, peptides, alcohols, vitamins, steroids, etc. All these can facilitate the growth of microorganism. Also, the environmental and formulation conditions favor the growth of microorganism. pH, oxygenation, temperature, superficial activity, osmotic degree and perfumes present in cosmetic products. The routine microbiological analysis of cosmetic products is based on determination of microbial quality of cosmetic product, and this includes counting of mesophilic aerobic microbes, most probable number of total coliforms, counting of yeast and molds, presence or absence of Staphylococcus aureus probe, presence or absence of Pseudomonas aeruginosa probe. The microbiological testing is carried out on raw materials to make the additives free from microorganism and also on finished cosmetics to prevent cross-contamination. Now first come to the sample preparation for microbiological analysis. For liquid type of cosmetic products, take 1 ml of liquid formulation which is diluted with 9 ml of modified lethene broth in screw cap test tubes. For solid powder type of cosmetic products, weigh 1 gram of test sample into screw cap test tube that contains 1 ml sterile 2-in-80. Then disperse the cosmetic product in 2-in-80, add 8 ml of sterile broth and mix thoroughly. Now come to the wax type of cosmetic products like lipsticks. Weigh 10 gram of cosmetic product into sterile 2-in-20, then disperse the product in 2-in-20 with sterile spatula to form a paste. Then add 78 ml of sterile broth and mix thoroughly. After sample preparation, come to the next step, that is inoculation. For inoculation, there are different methods like pour plate method. In this, the test product is diluted with sterile water. The agar medium is first sterile and is maintained in molten state at 45 degrees Celsius. 1 millimeter of diluted sample is mixed 9 millimeter of sterile cool agar medium, which is then poured into a sterile petri plate. The contents of petri plate are thoroughly mixed and then allowed to solidify. The petri dish is incubated at specific conditions of temperature and time. After incubation, observe the growth of microorganisms and determine the viable count. You can see the different steps of this on the screen. Next type of inoculation method is spread plate technique. This is similar to pour plate method, except instead of pouring, triangular type of loop is used for uniformly spread the sample over the surface of agar. Now come to the next type of inoculation technique, that is streaking. In this, the cosmetic test sample is diluted and the required quantity is transferred to sterile petri plate. The plate is inoculated with the culture of microorganism in a specific fashion of streaking by a special streaking technique that you can see on the screen. Then the plate is incubated at temperature suitable for microbes for a certain period of time. After incubation, the microorganisms are counted and calculate the viable count. Next method is membrane filtration method. In this, a weighed amount of pre-treated cosmetic material diluted with buffered NaCl peptone solution are given. Transfer this membrane onto the surface of solid nutrient agar medium in a sterile petri plate. Then this is incubated at suitable temperature for a specific period of time. After incubation, observe the growth and calculate the viable count of microbes. Next is serial dilution method, that is also a type of inoculation. This method is based upon the statistical probability. Cosmetic samples are prepared like spread plate method. Different serial dilutions are prepared and are transferred to different 9 to 15 test tubes. The number of organisms per gram are calculated using standard MPN, that is most probable number tables. According to the Indian standards, the bacterial count of any cosmetic product should not exceed 1000 microorganisms per 1 gram of cosmetic and should not contain any pathogens. The next test is microbiological challenge test for preservatives. That test is used for efficacy of preservatives. In this, microbiological challenge testing, predictive modeling and storage testing are the tools to test or to stimulate what could be happen to a cosmetic product during a shelf life, including processing, transportation, distribution and handling. Microbiological challenge testing is done if the microbes of interest is supposed to be present in small numbers or and incidentally come in contact with the cosmetic product. This test should be applied if full information about the potential hazards of microorganism is not available or reported. Microbiological challenge test involves several steps. First is the software-based experimental design, the appropriate inoculation method, then test procedure and interpretation of data. If the challenge test is done properly, then the actual information is found on the species or type of microorganism responsible for the growth in the cosmetics, so that risk of skin infections can be assessed. Therefore, the knowledge obtained might be the basis for setting the safety criteria at critical control points in a cosmetic product manufacturing operations. The preservative property of the cosmetic product is adequate if there is either a significant decrease or no increase in the viable count of microorganisms in the test sample of cosmetics after providing suitable conditions for the growth of microorganism. This test takes about 35 days for final conclusion about the result. Now come to the conclusion. Variety of additives or excipients are used for the preparation of different type of cosmetics. So the final cosmetic product when applied on human skin have the potential risk of types of adverse effects. The adverse reactions include allergic reactions, irritation, contact urticaria, sensitization, photoallergy, and phototoxicity. With growing awareness of consumers and implementation of Consumer Protection Act, it is important and necessary for the cosmetic manufacturers to assess the efficacy, quality, and stability of cosmetic product. Thank you.